Hi, I'm Professor Doe, Biology and Health Sciences Professor at Urban Valley College and Saddleback College. In this video tutorial, I will be showing you how I do 6 basic genetics problems. I recommend that you pause the video and take a look at the problems that are shown here on the screen and to try to solve them on your own. And once you're done with that, then push play and then I will show you how I solved this problem. So without further ado, let's go ahead and get started. Okay, so we have here our very first problem, and the problem here states what are the gametes that can be produced by the following individuals. And problem A here is big R, problem B here is big R, and problem C here is little r. First off, just a reminder that gametes are reproductive cells. And more particularly in humans, they are sperm and egg cells. Okay. And one of the things about gametes is that they contain 1/2 the chromosome count. And also, if the parents had 2 copies of a particular allele, a particular gene, I mean, for example, big B, then only one of these will go to the gamete, for example, big B only. So that's kind of how we would look at that. Okay, so let's go ahead and do the first problem. So part A here is big R. In order to determine which of the letters go into the gametes or what the gametes are going to have, what you do is you do something that I call the one gene split. So here's how you do so. Each of these letters here, you split them, and then you rewrite them down below. This is called the one gene split. All right. And then there's a secret step that I like to do. Are there any duplicate letters? And there appears to be some here. So I like to cross out all the duplicates and only leave one. So therefore, based upon our answer, in the answer to A here is just R, big R. That's a gamete that's produced. All right. How about part B? Well, we have here, according to our problem, big R, little r. So I'm going to go ahead and rewrite that down on my paper. I'm going to continue with the one gene split. This one here is big R. That one there is little r. Are there any duplicates here? No, there isn't. There's a big R and a little r here, and they aren't the same. One is an uppercase, the other is a lowercase, so we have big R and little r gametes. All right. And then part C, we have here little r. Again, to determine the gametes, I do the one gene split. Secret step here is, are there any duplicates? And there appears to be one. We have 2 little r's. We don't need them all. Cross them out. So therefore it's little r only. All right. Hopefully that you find helpful. For those of you guys who are wondering, what if you have 2 genes in here? For example, I'm just going to do a sub problem just to demonstrate this. Just a little letter D here. If I were to have big R, big Y, little y, how in the heck would you determine the gametes of this? I like to use a strategy called FOIL. So you use the FOIL method to determine gametes. And how I like to do this is I like to split the genes. So there are 2 different types of letters. There are the R's and the Y's. I like to keep those 2 separate. And here's how I determine the gametes using that FOIL method. First step here, get different colored pens. I like to take the outers first. So as you can see here, it's going to be weird. I can't do it. Right here, big R, the first letter of the first group to the first letter of the second group, right here. So one of the gametes here is a big R, big Y. And then I take the first letter of the first group to the second letter of the second group. Whoops. Right there. So it would be big R, little y. Can't quite see that there. And then I like to take the second letter of the first group and connect it with the first letter of the second group. As you can see there. So it would be little r, big Y. And then lastly, I like to take the second letter of the first group and connect it with the second letter of the second group. There we go. So we have here little r, little y. Are there any duplicates? In this example, there are no duplicates. So we keep them all on the list. So keep all the gametes on the list. No duplicates. All right. So this is called the FOIL method. And this is useful for dihybrid crosses, where you are dealing with two different types of genes. This one you can't quite see. That's little r and big Y. I don't have quite a pen for that. All right, let's do the second problem. Here we go. How many offspring, homozygous offspring, would be expected in every 4 offspring of the following cross, big Q, little q, crossed with big Q, little q? All right, so the first step here is I would typically define my alleles. Okay, but unfortunately for this problem, it's not defined for us, so we're going to skip that step. The next step here is to write out the cross, so I'm going to rewrite that on my paper just for good habits. And then I'm going to determine the gametes. That's how it all starts, right? If you're going to have any offspring or children, you got to make sperm or egg cells first. So we got to make gametes. So the first step I do is write out one of the parents' genotype. I'm going to start off here with the left. Rewrite that down. Second step here is to determine the gametes. How would I determine that? Is by using the one gene split, just like before in problem 1. So here I go. That's big Q. That's little q. Secret step. Are there any duplicates? There are no duplicates here, so we don't cross out any of the letters. Okay. So this would be the parent. Here would be the gametes. All right. And then we do the same thing for the second parent, which is the right one over here. Big Q, little q. I do the one gene split method. There. Little q. All right, same thing. These are gametes. This is the parent. Secret step, cross out any duplicates. There are no duplicates here, so we hang on to them. All right. Next step here. I need to do a cross, and I need to determine the probability of offspring. So for that, I need the Punnett Square. I'm going to move my page a little down here so you can see. All right. So I need to put these gametes on the Punnett Square, just like the multiplications times table that you've probably learned back in elementary school. Let's go ahead and get started with those. Let's take the first set of gametes from one of the parents. I've got 2 different types. I got a big Q and a little q. So I'm going to go ahead and randomly select them to go on the top of this square. So there are 2 places you can put gametes here, at the very top or on the very side right here. So what I'm going to do, I'm going to put big Q here, and I'm going to check mark as I go that I've already used that letter. And then I'm going to put a little q here because that was the next one. And I'm going to put a check mark to remind me that I've already used it. And I'm going to separate these 2 with a line. And do the same for the other parent. Right here, big Q for this guy. And the little q goes down here. And separate those with a line. Got 4 boxes. Just like the multiplications times table, we're going to put these letters together. Big letters always come first. That's a convention. So in this box, we have big Q, big Q. This one we have big Q, little q. Here we have big Q, little q. And then down here we have little q, little q. Just like the multiplications times table. All right. So it's asking for every 4 offspring the following, what would be, how many homozygous offspring would be expected in every 4 offspring of the following cross? Homozygous, several types. If we're talking about 2 big Qs like this, where Q is generally a dominant allele, this would be known as homozygous, dominant. If we have 2 tiny little q's or little letters, that would be homozygous, recessive. And if I were to have them different, where capital letters always come first, this would be known as heterozygous. Now our problem is asking just for homozygous, didn't really specify whether it was dominant or recessive. And so therefore, we can say, well, this one here is homozygous, and this one here is also homozygous. So to get the homozygous, would be 50% of the offspring. So therefore, if you have 4, it's expected that 50% of 4, 2 offspring, that is homozygous. Okay? All right. There we go. So that's the answer to this one. All right, let's go ahead and begin the third problem. Very similar here. How many homozygous offspring would be expected in every 100 offspring of the following cross? All right, we're going to do the exact same thing as we did in problem 2. If you need a review, just rewind the video back. Okay, so in doing problem 2 here, I'm going to go ahead and define my alleles. Well, unfortunately for this problem, they haven't defined the alleles, so therefore I'm going to continue on with the next step, which is to rewrite the cross down here. I love to rewrite the cross. All right, there's my cross. This was a mistake here. Okay, so the next step is to write down one of the parents' gametes. We have 2 of them here. They decide to have a kid. So I'm going to go ahead and deal with the first parent, which is big B, big B. Next step is to determine the gametes. In this case, I'm going to use the 1 gene split strategy. Just split these 2 letters, big B and big B. Secret step are to cross out duplicates, and in this example, we do have a duplicate. There are 2 big B's here. We don't need all the duplicates, so I'm going to cross out one of them. Again, this is the parent's genotype, and these are the gametes. Okay. Do the same for the second parent. And the second parent here is big B, little b. Next step is to determine the gametes. B, little b. Okay. Secret step, cross out any duplicates. We don't have any duplicates. They're all different, so therefore we hang on to them all. Right. So now we need to do a cross. That's what our problem is asking for. So that is automatically our Punnett square. All right. Put the gametes on either the top or the side, whichever parent you choose. I'm going to go and start with the second parent because I'm just randomly picking that. Start with the second parent, and I'm randomly deciding to put this parent on the top bar. So I'm going to say here, big B, check mark as I go to make sure that I have already used that letter and not make any mistakes, and little b here. So I'm going to go ahead and put a check mark there to remind me. And I'm going to separate these gametes with that line. Okay. Put the other parent's gametes on the other side. The first parent I didn't do yet, and the only gamete here is big B, so that's it. Put big B there. No line needed. Now determine the offspring. So we have here a sperm or an egg cell mixing up with a sperm or an egg cell that they're going to fertilize, and in this box we're going to have these two letters come together like a multiplications times table. Big B and big B. And the second box here is big B and little b. I often like to leave a little bit of space here. In a later problem, I'll show you why that is. Well, I like to use these to write down the phenotypes, but that's a later problem. Okay, the problem asks that for every 100 offspring, how many of them are expected to be homozygous? Just to kind of refresh your memory, homozygous dominant in this case would be big B. Homozygous recessive would be little b. And then heterozygous would be big B, little b. Always capital letters come first. All right. So the problem is, how many are homozygous? Well, in the problem, it didn't specify whether it had to be dominant or recessive. It just said homozygous, and it appears that these two is what we're looking for in our table. And according to our table, 1 is homozygous. This is not homozygous. So it's 1 out of every 2 are homozygous. But that doesn't answer the question. That just gives me the stats. Okay? It's asking for every 100 offspring. So if you have 100 offspring, then you would get 50 expected to be homozygous. Okay? And how I determined that was by doing a little bit of math, by saying I start off with 100 offspring, and then the stats here, I multiply that by the stat, which is 1 out of every 2. 2 total offspring, and 1 that is homozygous. Therefore, 100 times a half is 50. I hope that you found that helpful. Okay, let's go ahead and do the next problem. Okay. So R here is dominant for normal. Homozygous recessive produces cystic fibrosis disease. So consider the following cross and use a Punnett Square to answer these following questions. What's the probability that their first child will be normal? Second will be normal. Third will have cystic fibrosis. Fourth, be a carrier. And what type of dominance does this demonstrate? Same thing here. First step, let's define the alleles. Well, I have some on here, and I'm going to rewrite it on my paper so I can put all the information down on paper so my mind can focus on other things, especially during a test or an exam. I don't want to be worried about these little details I could get mixed up on due to the stress of a test. All right, so here we go. I'm going to define my alleles. We have here big R. And I'm going to say big R here is normal. Now I know that homozygous recessive, which is this little guy here, they're the same. That's homo, and zygous means the offspring, and they're recessive because they're 2 little r's. and I found out that this is cystic fibrosis disease. Okay? And since they're homozygous recessive, a recessive disease will only express itself if there are two little letters in there. So I can safely say that one little letter would also be the same as cystic fibrosis. It is a recessive allele, so I can say that. All right. So I have the following cross. I'm going to go and rewrite that cross down here. I'm going to split this up. Big R, and little r. That's my new cross. And I want to know what is the probability that the first child will be normal. Okay. So here we go. First step here is to write one of the parents' genotypes. Let's go ahead and deal with the first one here. That's big R, little r. Second step is to determine the gametes. I'm going to do the one gene split method. There you go. Next, are there any duplicates? Cross them out. Cross out duplicates. There aren't any, so we're going to keep them. So these are the gametes. And up one above are parents. Okay, that's a parent. Do the same for the other parent. The other parent here is little r. First step and next step here is to write out the parent's genotype. Next step is to determine the gametes. I'm going to use, again, the one gene split. Here are the gametes. Secret step is to cross out duplicates. We do have a duplicate here. We have two little r's. We don't need all the duplicates. Cross them out. All right, now we have our gametes. Time to put them in a Punnett square. Okay, make some room for my Punnett square there. Put the gametes on the side. I'm going to go ahead and do the first parent, big R, little r, the gametes. Write each of these on one of the two sides, this or that, and then split them with a line. Since I've already used these two gametes, I'm going to go and check mark them off my list to remind me that I've already used them. And the next one here is to do the same for the other parent, but on the other bracket here. So we have one little r. I'm going to go ahead and put that there, and then check mark to remind me that I've already used it. Next, combine these. Big R, little r, little r, little r. You can see, just like a multiplications times table. And now what I really like to do is to write out the phenotypes. That's why I left a little bit of space down here. So big R, little r, okay, big R, little r here would be normal. Okay. Now the problem didn't state what type of dominance this is, so I can assume classical dominance. Okay. And I only assume classical dominance if no other information in the problem is telling me otherwise. Okay. All right. So therefore, if I were in an example here, have big R, it would express normal phenotype. If I have little r, this would be an individual with cystic fibrosis. And if I had big R, little r, they would be normal. Now we have two individuals who are normal. Physically and phenotypically, they are normal, but one of them has a little r in it. So this case, this particular type of genotype would be known as a carrier. Because even though on the outside they appear normal, but deep inside their cells, they are carrying that little r right there. That's why they're called carriers. Okay, that little disease allele there, so they're called carriers. All right, so here we go. We go back to our square. What phenotype is big R, little r? Well, according to my chart, it is a normal. And I'm going to make a little note, carrier. And the little r here is an individual with cystic fibrosis. And I'm just going to abbreviate here, cystic fibrosis. Okay? All right. So question A. Probably the first child will be normal. Probability of the first child being normal, well, normal's right here. There's 1 of every 2. That means the probability here is 1 out of every 2, or 50%. All right. Problem B is asking what's the probability the second kid will be normal? Well, guess what? It's still 50%. My gosh, there's a strategy, and please do not cross out any boxes. I see a lot of students doing this. They like to cross them out, so don't do that. Okay? It's still 1/2 or 50%. All right, for example, what's a probability of getting a boy or a girl? It's 50%. So what's a probability that the first kid is going to be a boy? 50%. What's a probability that the second kid's going to be a boy? Well, just because you already had the boy doesn't mean that the remainder has to be girls, right? You can still have a 50% chance. So it resets every time. So you don't cross out boxes. So it's still 50%. Question C, what's a probability that the third kid will have cystic fibrosis? In our example, 1 out of every 2, or 50%. Question D, what about the fourth child being a carrier? We have here carrier, 1 out of every 2, so it's still 50%. This is an easy question. And then now, question E is asking what type of dominance does this demonstrate? This here we are looking at is classical dominance. Hopefully you found that helpful. Moving on to the 5th problem here. Big L is for straight hair and Big W is for curly hair. And John and Janice have several kids. One of them has straight hair, 4 of them have curly hair, and 2 kids have wavy hair. And they decide to have more kids. So what is the mother's genotype and father's genotype? What's the probability that their next kid will have wavy hair, and what type of dominance does this demonstrate? So it's a little tricky here. So let's go ahead and keep following the steps that we've done in the previous problems. First step here is to define the alleles. We've already have them kindly defined it for us in the problem, where big L stands for straight hair. And big W stands for curly hair. Now, what's really interesting is that in the problem, there was one kid that had wavy hair. And it appears that wavy hair is kind of like a meld between the two. This is curly, and this is straight, and then it's wavy. So it's a mix between the two. If you were to look at wavy, you can't tell it's straight, but you can't even tell it's curly either. Oops. It's just wavy, so it's a mix. All right. So they decided to have several. So actually, the type of kids that we have here, we have kids that have straight hair. So we have here, for straight hair, it has to be big L, big L. Big W here is curly. And then we have here wavy. And the only way to determine that is by big L, big W. That's wavy. So those are our genotypes that we have. Okay. So it appears that this couple has been able to produce all of these different types of, all of these 3 different types of phenotypes, and we're only doing this based on the scope of a non-majors biology class. Okay, so we haven't there aren't any other unusual types of dominance. We're just using classic learning and topics that are learned in non-majors classes. Okay, here we go. So big L, big W, big W, big L, big W here all give these different types of phenotypes. So we have to work backwards. We don't know what the parents' genotypes are, but we do know what kids they produced. Okay, and they produced all these 3 different types of kids. The kids have here big L, big W, and big L, big W. So if I were to look at the Punnett Square, if I were to look here, something else. Let's predict that we had both parents that had big L, big L. Okay, let's predict that. Well, then all these squares would be big L, big L, big L, big L. Okay, and so we wouldn't be able to explain where big L, big W, and big W come from. So the genotypes of the parents can't be that. So not that. So based upon our data, we can determine that L's and W's are present in the parents. They had to have gotten from one of the parents, so it's there. Okay, how about LW, LL? Well, if I were to do a genotypic cross of this, we would have LL, LW, LL, LW. But it doesn't account for WW. This had to come from somewhere, so this can't be the parent's genotype. So the only way to get a WW, based upon trial and error of doing the strategies, I'll get straight to the point, is making this a W. So by having the parents with these genotypes, we can finally explain how the kids and all their kids was able to get these 3 different genotypes. Here we go. We can see here LL, LW, LW, and WW. So the parents have to be these genotypes to explain how their kids were able to obtain all 3. Okay. All right, so I left a little bit of room on my Punnett Square. So big L is straight here. Big L, big W is wavy. Big L, big W is wavy. And WW is curly. All right, so question A is asking, what is the mother's genotype and the father's genotype? Well, the mother is big L, big W based upon this. And same thing with the father, is also big L, big W. All right. Question B is asking probability that their next child will have wavy hair. Well, according to our statistics, Punnett Square, it is 2 out of every 4 offspring will have wavy hair, or 50%. So the next child, 50% wavy hair. And then C, what type of dominance does this demonstrate? Well, this demonstrates incomplete dominance. And the reason why is because we have straight and curly, but the mix between the two is a meld. Okay, so if this were red, big L were red, WW were white, then therefore LW would be pink, a mixed meld between the two. Right, hopefully you found this helpful. All right, so here is the 6th problem. We have here, big L is for black color and big W is for white color, and LW is for black and white patches of color on spiders. A male with the genotype big L, big W, and female with big W decide to have many baby spiderlings. So question A here is the probability that the first spiderling will be black. Question B is the second spiderling will have patches of black and white. Part C, if there were 29,482 spiderlings, what's the expected number that will have patches? And then part D here is what is the type of dominance that this problem demonstrates? Let's go ahead and get started. All right, so first thing I like to do, again, I like to define my alleles. We have them in the problem. Big L here is for black color. W here is for white. And LW is for black and white patches. because I see here black and white patches, it already clues me in that it is a polka dotted Dalmatian spot-like condition where both the black and the white are being expressed. So that means if I were to look at genotypes, big L would be black. Big W would be white. And big L, big W again would be black and white patches. That means that this whole case here is codominance, because we have both the letters L and W being expressed in this LW genotype. All right. So we have here a male spider with the genotype LW is going to mate with a female spider, big W, and they decide to have many spiderlings. So I want to know what's a probability of the first and the second, and so on and so forth. So to do that, I need a Punnett Square. But first, I need to determine what type of gametes they're going to produce. So the first step here is I'm going to write out one of the parents' genotype, L and W. Second step is to determine the gametes. I'm going to use the one gene split. Secret step, delete any duplicates or cross out duplicates. We've got none. They're both different, so we keep them. All right. Do the same for the other parent. The other parent here is big W. Next step, determine the gametes using the one gene split. Cross out any duplicates is that secret step. We do have a duplicate 2 W's here. We don't need them all. We only need one of them. All right. Let's go ahead and do our Punnett Square to see what are the possible offspring. I'm going to go ahead and put the gametes on. I'm going to randomly pick the first one here, and I'm going to put these gametes on the top. I could do either or, but I'm going to go ahead and put them on the top and cross them out as I've placed them on the square and separate that with the line. Same with the other parent's gametes. This one is only a W. Place that here. And then combine them. L, W, W, W. Since I already indicated in the problem that L came first and W comes next, so I'm going to maintain that order, keeping the L's first and W's afterward. It doesn't matter unless the problem, actually because the problem here put LW in this order. So I want to make sure that I keep that order when determining the gametes. Not WL, but LW only because the problem gave it to you in that order. Okay? All right, so what are I like to leave a little space to determine the phenotypes. Big L, big W, according to what I know, gives black and white patches. I'm just going to put patches for short. And then big W here is for white. All right. So we got. Okay, so to answer the first part of the problem, it is what's the probability that the first spiderling will be black? The probability that the first will be black. Well, according to our Punnett Square, we have patches, we've got white, but there are no black spiderlings here. So the probability is 0 out of 2, or 0%. Okay. Part B asks, what about the second one having patches? So what's the probability of getting patches, or the second being patches? Well, according to our Punnett Square, we've got 2 possible outcomes. One of the 2 is patches. So it's 1/2 or 50%. C, if there were 29,482 spiderlings, how many are expected to have patches? Well, according to our probability for patches, it's 50%. So we want to take 29,482, 50% will have patches. that's not the answer to the question. I asked you what's the expected number. So I need to get my calculator and get 50% of 29,482. Here's my calculator. So we have to hear 29,482. We're going to multiply that by 0.5 for 50%. And that gives me 14,741 spiders with patches of black and white. And then lastly, part D is asking, well, what type of dominance does this demonstrate? Well, according to what we know, again, it's codominance. So the answer to part D here is codominance. All right, guys, I hope that you found this very helpful and educational, and after watching this video, I hope that you'll be able to solve any type of genetics problems that might come up in your future on an exam or in a class or whatnot. Thank you for watching. If you have any questions, let your instructor know. If I'm your instructor, again, let me know through the way that that's appropriate for the class. And I will see you in the next biology video. Take care, guys.